Thank you. So, all right, so welcome to this discussion section. I attended one of these last week, and I thought it was really nice. So one thing is we get to know each other and we see who is all in the program and who is here. And the idea is to present, I think, some ideas which then immediately lead to a discussion in something which is incomplete and something which I hope also we can maybe complete a bit more and make a more full picture. So my name is Thomas Hillen. Should maybe start with this. I'm at the University of Alberta in Canada, and I have been working on mathematical biology all my career, mostly on cancer modeling. So then I'm interested in how cancer spreads, in particular cells, how cancer cells move inside the body, how they form metastasis, and so on. What I like to do here, though, is last week, and Luca also mentioned the question, how to bring together these two fields of ecology and cell movement, because now we're in a position that in both sides, in ecology and in cells, we follow individual particles. We can follow cells, like neural quest cells in the embryo. We can, of course, follow individuals like whales and sea turtles and wolves, etc. And people on both sides use mathematical modeling, and they use similar mathematical modeling, but it's not always clear if we have the same methods on one side or the other. And that's what I like to try to form a picture. So what I have in mind is like a pyramid. So I come to the pyramid at the end, maybe, which can kind of inform how the models relate, and maybe what similarities we have between the different areas. Okay, so everything starts with an animal path. Okay, so an animal path is like an animal path, and no, I mean animal in a more general sense. So it could be a cell, could be also an immune cell or a cancer cell or so, right? And then we measure the spread, the spatial spread of these animals. Okay, and I would like to classify all this by the types of measurements which we can do, or measurements which people do. Okay. And now I take my notes. So, for example, if we see a path, or if we see many paths, or maybe many individuals, we can measure things. The first thing, we can measure something like a mean velocity. And if I say velocity, I mean speed and direction. You might hear from my accent that I'm German, and we only have one word, which is Geschwindigkeit. And this means speed as well as direction. And so velocity as well. Right? This is a vector which has a length, which is a speed, and it has a direction. Okay. Yeah, macroscopic drift offset. You can measure something like a drift. We call it macroscopic drift. And we can measure mean square displacement. And so these measurements are grouped together here. And as you see, this is not all, of course, we can measure, but these are like population level measurements, right? So we would measure this for the entire population. In particular, mean square displacement, if you just look how far did the wildebeest go in my model. Okay. But then we can also nowadays have more individual-based measurements, so we can measure individual velocities. We can measure turning angles and turning rates. Okay, and then these type of measurements, they first of all they describe the individual path, but then taken for population, they also describe the population spread. And then the last one would be jump probabilities. So here you just quantify given time intervals how they jump from one point to the next, and then write down probabilities for this. All right, so now we have measurements, and I think all of us maybe might be mathematicians. So we want to come up with mathematical models for this. And some of us, I know, like PDEs, and others of us like stochastic processes, or maybe some of us even like statistics. And we have models in either of these three directions. And I don't want to favor one over the other, but of course, I work on some more than on others, so I'm more expert in one thing than in another thing. But anyway, so I like to look at the stochastic side and the analytical side. And now the board has a disadvantage that they're not splitting up stochastic and analytic in two sides of the board. So please see this as a torus which wraps around. So then stochastic and analytic is right next to each other. Let's start here. So if I have the macroscopic measurements, mean velocity, mean square displacement, and so on, I go right away all the way here and look at drift diffusion models. Yeah, drift. Equations. So I'm not going to write down any equations, but it's a PDE for the population density. It has a drift term and it has a diffusion term. And the drift and the diffusion can be directly informed from the mean square displacement and the mean drift which we measure. Okay. There are other names for this equation, right? This is also called the Fokker-Planck equation. If it comes from a stochastic process, or a Former-Kolmogorov equation. And I write this down because mathematically they're the same things, but they come from a little bit different understanding. So how does this go on the left here on the stochastic side? These kind of measurements you can write down the stochastic difference equation. In this case, it's an on-sign Hillen process. Okay, and you can do Monte Carlo simulations of those. One box. And then the experts of you know, of course, that from an Einstein-Uhlenbeck process, you can also get back to the advection diffusion equation there. So then I close my torus. I can also here to these equations. Yeah, if you want to interrupt me or so, do that anytime.
So why you have mentioned specifically Einstein-Uhlenbeck? That
As an example.
just as an example.
As an example, right? Yeah. So there's something specific in mind. So. Okay, so let's go for the individual measurements. So now if we have measurements of the velocity, you can measure the velocity here, the turning rates, turning angles, etc. And we're getting more and more of these informations, in particular in ecology for animals. So, so then you would, on the analytical side, you would use kinetic transport equations. So these are hyperbolic PDEs, which have an integral term, and the parameters are exactly those: the turning angle, the turning rates, and the velocities. They go right into this mathematical model. Yeah. They're also known as velocity jump process. So that's the same thing. And from those, you can then again go to a macroscopic time scale, a macroscopic space scale, and come back to the distribution equations right here. So that's the quantifier scale. All right, and on the left, what we have on the left here, so then we use a stochastic differential equation where you also have a velocity parameter, which can vary stochastically. And these are called Landau equations. And you can also use the individual base modeling, of course. And also from these, you can then go around the circle and come back to the distribution equations. And this shows you already why the drift diffusion equations are so important, because using the appropriate scaling, this is kind of a common denominator where you always end up to. For the jump probabilities, again, on the analytical side, you then look at position jump processes. People call it the spatial master equation. So master equation is a balance of probabilities, right? So you balance the probabilities jumping left or right in 1D or jumping up or down or whatever in high dimensions. But that's the same thing. And one example where this is used is the step selection function. Okay, and this is a hot topic in ecology. So those of you who work on ecological modeling, the step selection function is a really hot topic that many people use based on animal tracks to come up with this kind of function. So maybe I should maybe follow you here. I've never seen this used in the context of cells. And then, of course, also from here, you can come back to the diffusion equations, which the key word here is moment extension. For localized clouds. So localized movement around. So that the jumps are more or less localized. On the stochastic side here, if you have jump probabilities, you would look at an individual base model and where then where you just simulate a whole bunch of particles jumping. Right. So, and then one little arrow up here, then we get to the diffusion models. Okay, so this is as far as I wanted to present. I find a schematic of this quite useful because I work in one thing but not in the other, but maybe get an overview of how things are related could be useful. But let me open the floor for comments or questions.
Yeah, I want to read a bit more about this step selection function.
Yeah, okay. We have the jump probabilities. So this is so that's a probability to jump from Y to X. And in ecology, people assume this depends on many things. So it depends on conspecifics. If you want to jump to find a mate, for example, or it depends on food. You want to jump to a location where it's food, or you want to avoid predators, etc. So here the idea is that this is proportional to first of all the distance function. It depends on X minus Y. So that's how far away is it, and it's more likely to be close. But then the important bit is the external features, and this is modeled by an exponential. And then it has a matrix, and they always call the matrix theta. So let's call it theta times Z and Y. Well, that depends on what in Y. And all Z contains all these features. So it contains the mates, the potential mates, it contains the food. It contains information about predators, hostile environment, etc. And then this beta gives you a weight. How these covariates influence the probability of jumping there or jumping away. And this exponential form here is what makes it into a step selection function. So this is how they use it. Okay, and that's not all. So this is then normalized. So that you get the probability density. And then maybe here, so Z are the spatial covariates. Right, so it could also be temperature. For fish, it could be salinity of the water or temperature of the water or things like that. So it's a form to specifically choose these jump probabilities.
So I just want to ask, so imagine instead of, this I can see that you take, I assume W could be a Gaussian, no?
Right.
For example, no? So because you have a random walker. Then, okay, so then you multiply. Suppose instead I write a master equation.
Mhm.
Like, let's make it discrete. So that then I put the heterogeneity myself on the master equation.
Yeah.
So how different is going to be the results? Because certainly it's going to be different. You are multiplying what you expect to be, let's say, as if there was no heterogeneity, and then you just multiply with that ZX1. So then instead, if I microscopically I put, whatever I think would be the microscopy there. So people have, there is a convenience in terms of the data, or it's just such a big assumption. You just multiply an exponential beta t z.
Yeah. So I see it exactly the other way around. So I think the animal does this first. So it decides where would I have to go? Where's my good food resource? Where are less predator? And then you multiply it by the likelihood to actually get there. Right. So if my preferred location is Oxford, not Cambridge, and I'm too far away, then I still might not be able to get there.
So that's the intuition behind using the product of.
Yes, fine. Yeah. So there are two decisions. Do you want to go there? And then am I able to go there? And then you kind of assume they're independent.
Yeah, I have another question. So actually, about those cases when the scaling is not appropriate, when you cannot go back from any kind of individual behaviors for any distribution or something.
Very good question. That's awesome. Yeah, you're right. Right. So for example, here in what I call the parabolic scaling, this doesn't work. Well, then you have to use this model. Right?
Yeah, but sometimes even this one is not appropriate. I remember seeing the cases when you compare the data,
Yeah.
haven't actually those statistics, and then it doesn't agree with either scaling. It could be, right?
Sure. Absolutely. I mean, it's just modeling, right? So it's of course not guaranteed. I don't know if you go in this direction, but maybe these anomalous diffusion and the lily walks and so on. So I don't know where I would put them in this scheme here. For example, yeah.
Yes, that's exactly the books and stuff.
Okay.
That's one, of course, one aspect. Also, when you look at the individual treatment, so there's a lot of approaches using time averages. So you have single trajectory analysis,
Mhm.
and it's not necessarily the same as the ensemble average, even in the limit, depending on the model. And so I'm actually following up on the question before. What happens when you have processes with distinctive change points? Right, I mean, it's animal tracking where you have something like correlated motion, these animal birds, whatever they fly long distances, but then they search and you have non-ergodic motion, diverging time scale, because they just sit and think or whatever they do. How do you kind of assimilate this in this kind of approach?
Yeah, so it sounds to me like spatial dependent rates. So just spatial dependent settlement rates or spatial dependent turning rates or so.
What if no finite rates?
If there's no finite rates.
Yeah, when you have diverging time scales. I mean, there is
Yeah.
indication, there's some recent studies about rather down now where they show that at least the kites they analyze locally, they have these power of waiting times.
Okay. Mhm.
Then maybe we need extensions of drifted or something.
For sure. So yeah, I can definitely see that. Yeah. Good. Yeah.
Right. One of the things we worry about with cells is that we can't image particularly frequently, then when we think about having any or many cases, they're observed from actual. Is that something we have to worry about with animals as well?
Like you mean the measurement interval and that you don't
Yes,
kind
the
of,
measurement interval impact.
absolutely, yeah. So I know that people are looking into this. Kind of finding an optimal tact or an optimal interval to measure them. Yeah. Yeah, so there's a third side to this pyramid. So I wanted to come to a pyramid. So that. Right, so here I start here with your measurements. And now let me try to draw this pyramid thing. And so now we have a kind of a hidden layer in the back. But so here's my analytical side. And then here is my stochastic side. And so maybe on the back, we should also look at the statistical side. I think in particular what you mentioned about measuring the autocorrelations along the paths and so on. This is, I think, first and foremost, a statistical information, right? And then we need to think about how we put this into models and maybe how we make the connection then around this loop or around this loop to analytical or stochastic models.
So Thomas, I mean, the fact that you separate analytical and stochastic is just, I wouldn't, but just because, but I know I was just curious.
You wouldn't, okay?
I agree with you.
So I was just curious, maybe in the math, I'm a physicist, so I don't know, maybe in the math it's more natural to think like this. I don't know, is that?
Okay, now I say something very dangerous, but I say it anyway. So I think on the
We want the dangerous thing.
we want the dangerous. So I think on the stochastic side, you throw away a lot of potential with nonlinearities. So on the analytical side, you can put on any kind of nonlinearity. You have Michaelis Menten reactions, you have logistic growth, or complex growth, whatever. But doing this on a stochastic side is really, really complicated because you worry about all these correlations and so on. So I think there's one big advantage on the analytical side to use nonlinearities. Okay, now you can kick me out of the room.
Yeah.
But on the other hand, you're right? And, even if we analyze these kind of models, we often do correspond.
No, just because you put the arrows there, you yourself put the arrows like
and I put it
right there.
Exactly.
So then, okay, yes, the simplest
It's
model, the one
pretty-
which
it's
are
pretty
analytical,
thorough,
no, but that's, yeah, I see.
Yeah, so right. It's actually not the impression I wanted to make that they are far separate. Yeah.
Yeah, probably I would create a pyramid where I put maybe measurement, model, and something else, and experiment, So it's
Okay.
interesting to see that you think that way,
Yeah, you're right. Sure. Yeah. But it's also not easy, right? It's work. Go from stochastic to analytical bank, this is work because we usually don't have the expertise, right? So I know my PDE stuff, but I know much less about stochastic processes. And other people have exactly the opposite, right? So even though the connection may be clear, but it still needs work for us. It still needs effort.
So Thomas, if I were to ask you, so what do you think the main difference okay, you said that you mainly work in cell biology, application.
Yes, right, yeah, right. Yeah.
So for the things that you have seen, what do you think is the main difference of those that work in animal application to animal ecology? And the theoreticians that work in application. Is there a main difference you can point to?
Yes.
It's much easier to make on animals than on studies.
That seems that we are realizing.
Yeah.
No, but on the modeling side.
I would have two answers. So the one answer is definitely the step selection function. So So almost everybody working on animal movement patterns uses the step selection approach. But then they never go this path. Right, so they do step selection function and then they do stochastic simulations. So there's a clear gap, I think, that people just don't look at the analytical side of things. That's one thing, and the other is, yeah, for cells, if you look at cell movement, cells are never alone, right? They're always in tissue, in an embryo or in a cancer, it's in a tissue, etc. So the assumption that you have independent paths is always wrong. So that I think that's another challenge for cell tracks.
So when you're for that kind of step selection function, the distribution equation, so it's just the localized kernel, which is, you know, at that point, I mean, the ecologists that are working with these
Right.
selection functions are using non-localized kernels as well in there.
As well, right? If you look at seed dispersal, you can even have fat tail kernels or so.
So is this kind of a bit of a kind of missing gap then in terms of derivation of continuum models, good continuum models to account for non-localized dispersal?
Well, we know how to do this, right? We can do moment expansions, we can use second or fourth order moments and so on. So I think from the theoretical side, we know exactly what to do, but it's not utilized in ecology. So who are the people here who works in ecology? Ecology
An
model.
application of the problem.
Yes, yeah. You for sure, I know that. So whales and sea turtles, that's ecology.
I'm sorry.
Yes.
I don't have it clear yet. So what is the question that I was trying to ask? What is because the measurements, for example, they could be something else, right? On the population level, also on individual dynamics. And I was always thinking that we choose the modeling technique or the type of model according to what type of data, what is that we would like to
Yes.
model. For example, there could be also any kind of spatial statistics, the spatial distribution or anything else, and we just choose accordingly. Is that so? What is the question? Are we trying to think how could we unify all of this, or try to look at it from a different perspective? Or
first one, no, and the second one, yes. So first of all, I fully agree. The choice of model should be given by the data, and this is the point I want to make as well. But the different type of measurements lead to different type of models. I don't want to try to unify it all, but I want to paint a picture how this all kind of fits together.
I guess you just very much just focus on movement. There's a lot of other things that are going on in the sector. And I guess it's just very much looking at the modeling that.
That's true. Yeah, right. Yeah, I also found this helpful in this context because we do come from different backgrounds. And it's I think it's good to appreciate where other people come from.
But I see some disconnection between people with background in mathematics or in background like myself in statistical physics. It's surprising to me.
So I think the backgrounds are different in terms of how you phrase the model, but if you think about it like along the lines you're doing there, we model similar things, but depending on the background, the way we kind of encode them is slightly different. And I guess some of them kind of points out how those can be related to one another.
Yeah. Yeah, I want to open doors.
Yeah.
So that the corridors. Open doors. Yeah.
Let me open another issue.
Good.
I mean, we have all this kind of, right? You know, this kind of models in mathematics. Mathematical terms are based on some basic assumptions, right? And so the big question is, Okay, are those assumptions valid in nature? For instance, you already mentioned that Boltzmann answers is questionable. Right?
Right.
Is one, it was answered before. I mean, if diffusion, do we always get the diffusion equations and so on? I mean, it is important to also to understand for all these kind of models that there are limitations from the practice to theories.
Yeah. But there's also the whole issue of inference, right, and model fitting to data. And I completely left this out here, but of course, at some point you have to do this, right, and see how well the model fits actually with data.
You know, I was still thinking about this step selection function. So I'm thinking that, so in the case of the animals, somehow, I mean, there is, given that in general, we think there is no feedback from, you know, the terrain, the animal, just the pause and quench disorder, and then choose the, so they're interested in finding out what, you know, that ZXY is, no?
You're right, yeah.
So, I mean, on the cell level, I guess, I mean, the whole, you really describe the whole dynamics, it's super coupled between, I mean, it's just, there was Marx, he was talking about angiogenesis, like, it was so complicated to see, but then it seems inevitable to do it. And so here, you just, I mean, there is no feedback, in other words, from the terrain to the individual.
No, there is, of course, also cells, the sense environment, right?
No, in the cell, yes,
Yeah.
but no, here, since you want to find out what ZXY are, you're not, you know, you just, maybe there is a reason why this is not being used in cell biology. Because you would have to, as I was saying, so you have to make the microscopy of it, and then you have to say, so you write the master equation for knowing that there is some heterogeneity there, so which is sort of what you see. So, well, this is what my perspective is seen in cell biology, also when there is complicated simulation, for example. Maybe that's the reason this is used there.
Yeah, that makes a lot of sense. So in ecology, you can measure the food sources, you can measure the prey, right? Which is not so easy in cell biology. Yeah.
We use that selection function, and if I have, I think you have to do image kind of frequently.
You mean directional persistence?
Yes.
Yeah. Oh, it could. Yeah, I forgot this. So there could be another term. So let's call this, I don't know, G or theta, which is sometimes used and which contains a directional component as well. Basically,
So I guess a question for this study, selection function, right? Used often in ecology, is it just that, I mean, are there no cell types that we're aware of that would satisfy that? Or are you aware of some cells that maybe we should be applying that?
I think in principle it could be applied to any cell.
Any
Yeah.
cells?
Right, I have to say, as long as we can get information about the spatial distribution of covariance of interest.
Change over time, like the TFS biology, you just find it before you start running the model and then just run. It says the quality of gap from X, Y always the same for full time.
No, it isn't, right? This can still be time dependent. This can also depend on a different population, like a prey population or so, right? I wrote it down in the simplest way, but this can depend on many things. Anything else? So we can keep this in mind as a discussion.
Yeah.
For the entire week or next week as well.
No, for 6 months, it's only
Oh,
5.
6 months, okay, that's fine.
Yeah.
And then we revisit again and we complete the permit.
Perfect. Okay, thanks, Tom.
Thank you very much.